Okay, number 2 on our quick videos looking at the 5 assumptions related to Hardy-Weinberg equilibrium. One of them we talked about last week, which was natural selection. You just maybe watched a video about non-random mating, which all by itself is not an evolutionary force. And now we're talking about genetic drift. This one's really pretty simple, but it's kind of a weird one to understand. Because we're talking about random changes in allele frequencies. So the definition would be a change in allele frequencies that's due to chance or sampling error. And what that term sampling error gets at is that there are many individuals in a population and many alleles, maybe for a locus, and the rate at which all individuals reproduce is probably not the same. And so it's possible that allele frequencies might change from generation to generation based on who gets to mate and have nothing to do with the alleles that are actually getting passed on. It might just happen that some individuals with a certain allele are more likely to reproduce and that allele frequency increases in the next generation. So you could think of this like flipping your coin, and we know that the math says if we flip our coin 2 times, we're going to get heads once and tails once. But you know that sometimes you get 2 heads and sometimes you get 2 tails, and that doesn't mean that there's anything wrong with your coin or with your flipping ability. It's just random chance that sometimes you don't get an accurate sample of the alleles. All right, so that's genetic drift. Change that has nothing to do with the fitness of the allele, and that's just due to chance. We saw one type of genetic drift at the end of the lab with the beads, where you're just pulling out a few beads out of your bucket. Okay, so we call it genetic drift because it causes allele frequencies to sort of increase a little bit, decrease a little bit. They just sort of drift around. They're not going in one direction or another direction like we might see with selection. This is especially prevalent in small populations. So I mentioned flipping our coin twice, and we think it's probably pretty likely that we would get heads twice or tails twice sometimes when we flip our coin twice. If we flip our coin 1,000 times, we think it's pretty unlikely that we would get all heads or all tails, right? So the more we flip our coin, the more individuals that are in our population, the more matings that occur, the more likely we are to get a ratio that's close to whatever we started with. In the case of our coin, that'd be 50-50. Okay, so key points about genetic drift, and I always get ahead of myself, so I've kind of mentioned some of these already. One is that it's random with respect to fitness. So the changes in allele frequency have nothing to do with the phenotypes that are caused by the alleles. Most pronounced in small populations, and this is a really big concern for conservation. A lot of threatened, endangered species have small populations where they are likely to lose diversity just by chance. And what can happen with genetic drift is loss, or what we call fixation of alleles. So we can go from having many alleles at a locus to just one, and then genetic diversity for that locus is gone from our population, unless we have a mutation to give us a new allele. Okay. So 2 depictions of modeling outcomes. We've got the frequency of one of our 2 alleles on the y-axis and time on the x-axis. Figure A on the left is a population that's really small, so like maybe we're flipping 4 coins. And on the right, we've got a large population, so you can think of it like 400 coins that we're flipping. And so what this is showing you, again, illustrating that in a population that's really small, we have a much higher likelihood of fixing one or the other of our alleles and then losing the other one. In the larger population, You can see allele frequencies drift up and down. The colored ones are sort of the average of all of these model runs that are in the lighter color. So allele frequencies drift up and down, but at least as far as we've modeled out here to 100 generations, we don't lose one or the other of the alleles. Right? So random changes in allele frequency. We're going to look at two events, two phenomena that cause genetic drift. One is founder effects. We'll have a slide. And the second one is a population bottleneck. Right, so founder effects are related to colonization of a new effect. A new area. So founder effects. Who founds the population? So here in our box on the left, we've got our three, and this is modeling genotypes, right? Because alleles don't float around by themselves. Individuals with their genotypes bring their alleles. So what this is showing you is that our big population, out of our big population, a few individuals come and found this new population. And it just so happens that we get two individuals that are homozygous for allele 1 and one heterozygote individual. So now, out of the 6 alleles that are here in these individuals, 5 of them are the A1 allele, and we just have one A2 allele, which is really different from the population we started with that I think has 50-50. Okay, so the process, I got ahead of myself, the process of founder effects, immigrants establish a new population. And it's possible that just by chance, not because their phenotypes made them more likely to leave or made them more likely to survive the journey, just by chance, the allele frequencies in the founding population might be different from those in the source population. Okay, so there it is. This change in allele frequencies is called a founder effect. We see this with things like colonization of islands. We talked about that when we talked about Darwin. It's likely to occur with any founder event, even if the founding population is more than 3 or so individuals. Okay? The other force of genetic drift is a population bottleneck. This is a sudden loss of individuals, so a decrease in population size. This could be caused by a disease that kills off a bunch of individuals, maybe a natural disaster, a hurricane, a volcano, lava flow, something that causes high mortality, that kills a lot of the individuals in the population, but doesn't kill them with respect to their phenotype or genotype. So just like with our founder effect, it's possible that by chance, later in time, after this bottleneck event has occurred, the individuals who are left might have different allele frequencies than the population as it was before. Okay, so here we have, I think the blue was homozygous allele 2. So now I've got 3 homozygous allele 2s and 1 heterozygote. Okay, so 6, 7 recessive alleles and just 1 dominant allele. So a big change in time 2 relative to time 1. So we call this event a population bottleneck or genetic bottleneck, a sudden reduction in the number of alleles in a population for a particular locus. An example, this occurred in a human population on the Penjlap Atoll, which is in sort of the southeast South Pacific. So in the 1770s, a large population of over 1,000 people was almost totally wiped out by a couple of natural disasters, a flooding, hurricane, the whole thing. Only 20 individuals survived, and it just so happened that one of those 20 individuals had this disorder called achromatopsia, where there's no pigment in the iris of the eye. So this is really rare in the larger human population. Less than 1% of people have this, have both 2 recessive alleles. One individual in this surviving 20 had it just by chance. It didn't help them survive. It just happened that they had it. And so now, in the population that has rebounded after this disaster, more than 20% of the alleles in this population are the recessive achromatopsia allele. Right, so this doesn't just happen with colored circles, it really happens in real populations. All right, our next topic will be mutation. See you then.